Okay, good morning, students. Am I audible?
Good morning, ma'am.
So I'm audible, right? Few students are still joining, I guess. I can only see 13 students. Okay, so I think we can start and they can join in between, right? So last lecture we discussed in general about plant tissue culture, that what is plant tissue culture, certain definitions, certain principles of plant tissue culture, and aseptic conditions that are important in plant tissue culture. So we'll just quickly revise what have we done last time and then we'll move on to today's lecture, right? So who will tell me what is the basic principle behind tissue culture that is being exploited in growing plants in artificial conditions? Yes, no one? Okay, Manaswani.
Good morning, ma'am. So, the basis of plant tissue culture is that a particular part of the plant, so a particular tissue from the plant is taken and it's grown in optimum conditions, which basically allows for the production of, genetically identical clones of that particular plant. So, this is important and it's used because it can be used to prevent or treat viral infections because the
Mm,
viruses don't affect the meristem. So this is just an example. So the
okay.
meristem will be taken and using plant tissue culture, the plant can kind of be grown again and it will be virus free. So that is
Mm.
the basis of
Okay,
plant tissue
so
culture.
that's fine, Manaswani. But anyone else? I just wanted to hear about the basic concepts we discussed that time, right? Some basic. Yes, Shakti, can you please?
Yes, ma'am. Good morning, ma'am.
Good morning.
Yes, ma'am. The thing that is exploited in plant tissue culture is the ability of plants to survive in outer environment.
Mhm.
So, we
Yes.
provide them the exact requirements that they require, and then they have to learn how to survive in outer environment. So,
Okay. Anyone else? I'm still not getting the exact answer that I wanted to hear. Anyone? Yes, Devanshi. Shivani, you were saying something.
Ma'am, isn't it like growing a transgenic plant for a certain gene of interest, or growing from a
No,
particular
no. Okay.
tissue?
See, students, I'm just asking how this is possible. How this is possible? We are taking just a part from some grown-up plant and we are putting it, providing some media, and it is developing a new plant. So what is that? I wanted to know what is that, right?
Totipotency.
Yes, Namrita, totipotency. See, this is we discussed that time also that the basic concept is totipotency, redifferentiation, and these things. But the basic is totipotency, which means it is the any in potential of any living plant cell. So any plant cell can develop into entire organism. So any part or small part from a plant cell taken, even if it's a mature cell, totally grown up cells which are not dividing further or mature, totally mature cells, we just take a small part from that. So plants, they have the ability that if provided the proper conditions or a nutrient, they can develop into the whole plants. This is not possible in animals or humans except stem cells. We are not talking about that. So this is a special ability which only plants have, this thing, right? So this is the basis of the tissue culture. So these are the basis of a tissue culture, right? How a single cell can differentiate into many other cell types, right? So this is found only in a living plant cells, not in animal cells, exception stem cells. And this term was coined by the Morgan, right? So this is how during the tissue culture we just take an plant and from a differentiated mature tissue, which is already differentiated and mature. So it undergoes a process, right? We take a explant, it undergoes a dedifferentiation, it form a callus, which is undifferentiated mass. Then it goes again redifferentiations into the new plantlet. So totipotency is the basic property in the plant that we are exploiting, right? So if someone asks you the basics of a plant tissue culture, you're going to talk about potency, right? That term I wanted to hear. Yes, another important thing. So, what are the important requirement for a plant tissue culture? What are the common things? Just enlist those things. What things we need for a plant tissue culture? No one raising hand, so I'll randomly ask, right? Yes, Yukta. Yes, Yukta. I hope I'm audible.
Ma'am, excuse me.
Yes, Devanshi.
Ma'am, actually, I wasn't able to raise my hand. I don't know why.
I don't know. Last time also you guys were not able to raise your hand, and this time also
Yes,
again you are
ma'am.
saying.
Can I?
जबकि मैंने आप लोगों को किसी को भी शायद अभी वो भी नहीं बनाया है. You all are just team members. So
Ma'am, I don't know.
Can
My
I
team
answer?
isn't responding. Can I
okay,
answer this question?
okay, fine, go for it.
Ma'am, basic things required like media, it can be MS
Yeah.
media, gel agents, agar, containers, and for maintaining pH meter,
Mm-hmm.
ma'am.
Yes, Shivani. Okay, Devanshi.
Ma'am, sterilized conditions,
Yes, that is most
nutrient
important thing. Yes,
nutrient medium for
yes.
growth and development.
Yes, yes. So sterilized conditions and a proper lab where proper aseptic conditions can be maintained is a very important part of a plant tissue culture. So whenever we are doing the subculturing or a tissue culturing, we need to make sure that we are doing it in a laminar flow. We are using lot of 2 to 3 sterilizing agent to sterilize the explant. Then only, and in the media also we add certain antibiotics to prevent any kind of a bacterial growth. So a lot of things, a lot of care is being taken, right? Okay, so today's lecture we are going to start about today we are going to discuss about a topic which is called somatic embryogenesis, right? So before that, let's see what is embryo. So I hope, see, in a plant, you must have studied all things in a plant science 1 or a plant science 2, right? So those are the terms. Since it is a plant tissue culture, so we'll be using lot of terms like embryo, might be using lot of times somatic cells or a zygote or a fertilization. So वो वाली सारी terms इसमें हम बहुत commonly we are going to use, right? So embryo, we all know, is a part inside the plant seed which has from embryo plant developed, basically, right? So in a short form, from embryo plant is being developed. So seed is the starting point for the plant. As the embryo, it activates in the right conditions and eventually it becomes the seedling, right? So what is fertilization? We all know, right? Fertilization could be asexual fertilization in a plant also, and a sexual fertilization. So it is a process where pollen fuses with the ovule to form an embryo. This is a sexual fertilization, right? So this is an important step in plant sexual reproduction. So when fertilization, after fertilization, zygote is formed. We all know that, right? So this zygote undergoes lot of cell divisions and it forms a embryo. Now this process of developing an embryo is called as a embryogenesis, right? This is just a repetition. So parthenogenesis is another term, right? So when we talk about somatic embryogenesis, then we should know about parthenogenesis also. So this process occurs in some plants, natural process which occurs in some plants such as pineapples or bananas. Here the fruit is formed without going through fertilization. So you say, how even this is possible? There's no fertilization and fruit is being formed. So the female part of the flower directly converts into a fruit. So the fruits which are developing, they are seedless here, right? So in different researches also, it has been observed that tissues that are part of the embryo sac or those surrounding it can develop into the embryo, right? However, proper environmental conditions or what are the requirement for development of the embryo are being required. So this idea led to the development of somatic embryogenesis method using the same principle. So same principle is being exploited, and in a plant tissue culture we are using that to follow a technique called somatic embryogenesis. And in the last, we are going to discuss what are the applications of this somatic embryogenesis. So see, in this course, we are going to study different techniques. And in many different techniques, you'll find at least 2 or 3 applications are similar. That somatic embryogenesis से भी आपको पैथोजन फ्री प्लांट फूड प्रोड्यूस होंगे, मेरिस्टम टिप कल्चर से भी होंगे, और कोई मेथड्स भी होंगे जिससे होंगे, राइट? सो यस, there are certain common uses of many techniques of a plant tissue culture, right? So parthenogenesis, okay, this we have done. So somatic embryogenesis is basically a process where a single cell or a small group of cells, they follow a developmental pathways that leads to reproducible regeneration of non-zygotic embryos. So I'll discuss later what are zygotic and non-zygotic embryos, which are capable of producing the entire complete plant. So these non-zygotic embryos, they may originate directly from other organs or parthenogenetic embryos. Without fertilizations जो embryos develop होते हैं उनको हम non-zygotic embryos बोलते हैं, right? Or androgenetic embryos which develop from the male gametophyte only. In general, these somatic embryos are those which are formed from somatic tissues in cultural or in vitro conditions. So embryos which are formed in cultures, they have been designated by various names, maybe accessory embryos or adventive embryos or embryoids or supernumerary embryos. So they are not called zygotic embryos, they are named by the different other names, right? So the initiation and development of these embryos from somatic tissues in a plant culture was done by Stewart et al. In 1958. And some other people in some plants, right? So somatic embryos are formed by sporophytic cells except zygote, directly arising from other embryos or organs, which are termed as adventive embryos also. Parthenogenetic embryos are formed by unfertilized eggs. Then we have androgenetic embryos. These are, as the name suggests, these are formed by the male gametophyte, such as microspore or a pollen grains. So somatic embryos should closely resemble their bipolar structure as in case of the zygotic embryos. They should be appropriate root, shoot, and cotyledonary development, as this happens in case of a normal zygote also. So from this also, the same process takes place, right? So if we talk about somatic embryogenesis, it is of 2 types: direct embryogenesis and indirect embryogenesis. So when we talk about direct embryogenesis, so embryos, they initiate directly from the explant, right? So we know what is explant, right? Explant is that part of the plant which is taken to further grow in the tissue culture. It could be the root tip, it could be the shoot, it could be the meristem tip, it could be the seed, it could be the pollen grain or whatever. Plant का जो भी part हम further full plant को grow करने के लिए लेते हैं tissue culture में, that part is known as explant, right? Remember, this term is going to come again and again. So the embryos which initiate directly from the explant without callus formation, and here some cells which are called pre-embryonic determinant cells, PEDC, they initiate embryonic development. So only those cells are required to be released. So such cells are found mostly in the embryonic tissues, certain tissues of young in vitro grown plants such as hypocotyle, mucellus, embryo sac, etc. So all of them, they come in this, right? And then we have an indirect embryogenesis. So here what happens, the embryos, they develop through cell proliferation. That means in indirect embryogenesis, the callus formation takes place, right? So in direct, no callus formation takes place. In indirect, the callus formation takes place, right? The cells from which embryos arise are called induced embryogenic determinant cells. Here, growth regulators with specific cultural conditions are required for initiation of the callus and then redetermination of those cells into the embryo development. So certain proper nutrient conditions are being required for the so that the callus can be further developed into the embryo. Then somatic embryos, they arise from single cells located within the clusters or meristematic cells, either in the callus mass or in suspension. Such cells, they develop into pro-embryos with polarity, which follow the same pattern or mimic the general pattern which is associated with in vivo embryos in the ovule, right? So pro-embryo initials may be single cells or multicellular groups. When the conditions are suitable, these embryos, they will germinate to produce the plantlets or a smaller plants. So this shows the mass production of plants through somatic and secondary somatic embryogenesis, right? Or proper culture, how callus is being initiated, how callus is established, and how there is a cell suspension culture, right? And then germination is being taking place, right? So this is how it is the process it goes on. Then since we all are talking about the plant tissue culture, right? Somatic embryogenesis is also we are doing through tissue culture. So definitely a medium or a solid media or a liquid media with specific growth regulators are the requirement of this particular technique, right? So this definitely there are different stages from callus initiation to embryo development, then maturation, and then plantlet formation. So this I've discussed in the first lecture also, that though we know the basal media or a basal MS media, or maybe there are already thousands of media being discovered by different people. But when we are working on a certain new plant or for our research purposes, so we need to standardize, right? We need to standardize, or we need to see that what kind of a medium is required. So for many species, different types of media are required for the whole process. So we know the function of different growth regulators, right? The role of different growth regulators in the growth of development of the plant. So similarly, when we are growing a plant in tissue culture, so we need to add certain hormones in a proper concentration, right? So presence of auxin is important for embryo initiation. But then if we lower the auxin content further, it is helpful for the embryogenic clump formation, which ultimately develops into the mature embryo. So how much concentration is required, that again we need to see, right? In general, it has been observed that auxin or auxin in combination with cytokinin in different ratios appear essential for the onset of growth of somatic embryos. Sometimes the medium which does not contain auxin, but certain level of cytokinin or abscisic acid may also prove beneficial for the somatic embryo. So these statements are because some people have already done research in this area, and they have seen that such variations or variations in the different level of hormones how they can impact the result, right? So these are the research outputs. So sometimes the addition of this is important. So we add charcoal into the medium, and they say it is important or it is useful for somatic embryo development. Can anyone tell me why charcoal or activated charcoal or how it is helpful in somatic embryo development or in a plant development in that by using this in the media? Any idea? Any vague idea? नहीं? This is your assignment. So आप ये कल पढ़ के आओगे और मुझे बताओगे next class में why activated charcoal is being used in the media. What is its importance, right? So I'm not going to tell you this.
Ma'am,
Yes.
ma'am, isn't it at least whenever we've used activated charcoal in any other experiment, it was used to remove impurities because the impurities get adsorbed on its surface.
Yes, Manaswini, you're there. This is used to remove the impurities or some compounds which are not phenolics or certain other compounds which are being released and which are not good for the growth of the plant. So yes, these are charcoal has a property adsorption, right? It adsorbs things. So this is how it is being used. Yes, good, Manaswari. So what is difference between zygotic embryogenesis and somatic embryogenesis? So we know now what are zygotic embryos and somatic embryos. So in angiosperms, the ovule contains haploid cells or ovum, which is a female gamete. So it gets fertilized by the male gamete, resulting in the formation of a zygote. So this is a process of a fertilization. So this zygote gives rise to multicellular embryo, which is known as zygotic embryos, right? So those embryos which develop from the result of a fertilization of a male and a female gamete are called as zygotic embryos, right? And this process is called as a zygotic embryogenesis. Then embryos can be formed as we know, right? Embryos can be formed from unfertilized egg or any other cell of a female gametophyte also, or the sporophytic tissues. These embryos are called as a non-zygotic embryos. So in vivo, there is no embryo development from any somatic cells, right? That is, the somatic cells does not have the embryogenic potential in general, but we are exploiting this in a artificial conditions, or artificially we are doing this process, right? So, but due to the totipotency nature of the plant cells, the somatic cells having the complete set of a genome may be induced in the cultures, so we can do it artificially to form the organized bipolar structures which has the cotyledons which resembles the zygotic embryos, right? So we can do this artificially this process can be done. Now this process or this phenomena is called as a in vitro somatic embryogenesis. So term embryoids are sometimes used to distinguish the somatic embryos from the zygotic embryos, but it is fine. We use the somatic embryos or somatic, these terms can also be used. So any sporophytic cell or a gametophytic cell may undergo somatic embryogenesis due to some changes which occur at the molecular level, that is maybe induction of some genes or a protein production or some high kind of a metabolic activity, so it can be done, right? So the stages of the development process of both somatic embryo and zygotic embryo, they resemble each other morphologically also, so they are similar. So in both the cases, the single somatic cell, sporophytic or a gametophytic, or the zygotic cell, it undergoes divisional phases to produce a globular stage, torpedo stage. We know the different stages of a zygote formation, all those things, right? So it has a globular stage, then it has a heart or a torpedo stage, right? So at the end of which cotyledons or first leaves, they begin to develop. So these are the similar in both the cases. So in dicots, 2 cotyledons, in monocots, 1 cotyledon emerges. So after this stage, the developmental process differs in zygotic and somatic embryos. Up till this stage, the process is similar. So these are the processes of zygotic embryo development and somatic embryo formation and its development, right? Diagrammatic representation. So the zygotic, there are certain differences, right? So in zygotic embryos, they undergo prolonged maturation phase, right? And many storage proteins are being synthesized during this time, which is primarily regulated by the abscisic acid. So after that, dormancy period is required for further germination. But in somatic embryos, we don't need a this dormancy period is not required. So somatic embryos can differentiate and grow to produce the root and shoot with leafy development without any dormancy period. So this is the difference here, right? Another important morphological or developmental difference is that somatic embryos, they are devoid of suspensor and the endosperm, which is present in the zygotic embryos. So unlike zygotic embryos or a seed embryos, the somatic or adventive embryos, they are devoid of endosperm as a food reserve. So it is not, this part is not there. So fluid drilling devices are sometimes harnessed to sow the naked embryos directly to the soils, or these embryos are being encapsulated in some plastic strips or pellets together with small amount of a nutrient. So aimed at using embryos for a large scale planting. So this is how they are going to do it artificially, right? Now coming to the application part of this process, that why actually this process is important in a plant tissue culture, right? So first application is a large scale propagation compared to zygotic embryo. So if we compare to the natural process of a zygotic embryo, the large scale propagation is possible in case of a somatic embryogenesis. So induction of a somatic embryogenesis forms the ultimate goal in free cell suspension cultures. It again rely on the totipotency of the cells and could reasonably be exploited for the micropropagation, right? So micropropagation is another small technique in a tissue culture by which a number of plants can be produced, right? So each cell of the suspension cell culture can be induced to produce somatic embryos, which can be maintained in an arrested state of a development by cold storage. We can just do some cold storage process and they'll be remain in that stage until the time of the sowing. So somatic embryogenesis is highly desirable and holds out promise for rapid multiplication in a shorter time with the shoot root axis. So rapid multiplication of the plants in a smaller time can be done by using this process. Then next we have a more useful than organogenesis. So mass production of advantageous embryos in a cell culture is still regarded as an ideal propagation system, right? Propagation का मतलब होता है to grow the plant further, multiply the plant. The advantageous embryo is a bipolar structure which develop directly into complete plantlet, and there is no need for a separate rooting phase as with the shoot culture. So directly it plant, we can develop into the plant. We don't require a separate shooting phase or a rooting phase here in this. Then useful for mutagenic studies and mutant productions, right? So mutagenic studies are basically, see, if we want to study that, if some kind of what are the impacts of certain mutations in the plant, how does it affect that? So those studies can be done here, because somatic embryos, they generally arise from the single cell. So it is advantageous for mutagenic study. Also the plantlets which arise from somatic embryos, they are homogeneous, similar honge, right? Because when we do these kind of mutagenic studies, we don't want changes, because एक हम control पे रखेंगे और एक में हम mutations induce करके will check what are the changes, right? So this technique is more suitable, right? Because they are more homogeneous in nature. So mutant gene expression can be studied well. So we have a similar plant. What are the gene expression? How genes are being expressed if there are certain mutations in a certain genes? Those kind of studies can be done. Expression of a particular gene can be studied here in a much better way or a much easier way. Then useful for genetic manipulation. So genetic manipulation is, again, we are manipulating the genome of an plant to maybe for the yield increasing, maybe for the resistant to a pathogen, or for any other reason, right? So in a plant biotechnological applications, during any foreign gene transfer, if transformed cell give rise to plantlet by a somatic embryogenesis, there is a least possibility of chimera formation. Chimera formation होता है जब maybe in there's a different genetic in one plant, the it might have a genetic composition from other sources also, a chimera formation. So that possibility is ruled out here, right? So for transgenic plant production, this method of multiplication system is very much useful. Then we have again, ये जो pathogen free plant production वाला है ना, ये आपको हर जगह दिखने वाला है. So useful because plant tissue culture is a technique for a pathogen free plant production. So useful for pathogen free plant production. Plants derived from this kind of a somatic embryos, they may be free from viral or other pathogens also. So we can use this as alternative approach for a disease free plant production. So that we know, right? Then a good source of a protoplast culture. So after this somatic embryogenesis, we are also going to study about the somatic hybridization. So these are two different technique. So in somatic hybridization, there is a protoplast fusion and protoplast isolation, all those techniques that we are following, right? So it is a good source of a protoplast culture also. So embryogenic cultures are especially valuable in providing a source of regenerable protoplast in gramineous or some coniferous plants. So protoplast from these cultures were induced to divide to form a cell mass from which the embryoids or even plantlets can be regenerated. So this you're going to study in the next technique that we are going to discuss, right? And then conservation of genetic resources. So conservation, you'll see that why conservation of genetic resources is important. So I'm sure in a bioresources and bioprospecting course you have studied last semesters, and in that it was there was one topic that why we need to conserve the genetic resources, right? It is why. So that big question was there, that how we are exploiting our genetic resources, how the resources are less now. Maybe we have overexploited them for the medicinal value or any other thing, right? So that's how we need to conserve them right now. So somatic embryos which originate from a single cells and subsequently regenerate mostly genetically uniform plant, right? Changes nahi hai, uniform plant hai. We need to maintain the germplasm also, right? Genetic resources. So we don't want any changes in that. So these are good material for genetic resource conservation also. So embryogenic cultures as well as the somatic embryos, they remain viable upon storage at a proper temperature or a cold storage or a cryo storage, and they can be further used when they are required in further studies. So this is, we can conserve them, right? So there are germplasm banks all over. We have in India also, we have at the international level also germplasm banks. So whenever we are working or whenever we are looking for certain germplasm which is not available, or a germplasm which we want a original one. We don't want any kind of a genetic changes or a changed one. So we look for these germplasm banks where they are being stored to use them or to get back them so that we can work on that, right? So these are important to store in the germplasm banks. So advantages we have discussed already. So higher propagation, maximizing output. It is suitable in suspension culture and also for large scale production, which involve bioreactors also. So certain secondary metabolite production, all those things, we can go for this, right? Artificial seeds can be produced. Germplasm conservation, as I told you. Then seedling can be pathogen free. This we have discussed. So this method also permit easy scale up and subculture with a low labor input. So it is economic also. But still, we have with every technique, definitely it always comes with a certain limitations also, right? If we have many good points, then we have a few limitations also with every technique involved, right? So there are certain limitations. Response of somatic embryogenesis is a tissue specific, so they have lot of studies have been done that they shows that success depends upon the type of the explant we choose to work with. So response can be differ or it can vary with the change in the explant or a type of explant we are using. So only certain explants would produce expected result. Not all are giving so much good result. So this we need to see that, right? So there is a high chance of low frequency of embryo production. Also, there are chances that ये हो सकता है, right? So many time कुछ embryo incomplete embryo production भी हो सकता है, so that can be again a drawback, right? Then there is a high probability that sometime this method can lead to some genetic variations also where we don't want any genetic variation. So कुछ chances, percent chances, they are always there, right? So in some plant species, this method is unable to generate large number of normal free living plantlets, not visible everywhere. And in some plant species, the plants developed by this method might be weaker. They might not be able to survive in the natural environment later. So they have definitely with the good things, there are some negative points also, but still this technique is being used and it is being widely used for a lot of purposes. So it's not that just because of few drawbacks we are not going to use that, but we need to be careful. So we need to be very careful when we select the explant. We need to be very careful, right, where we don't want any genetic variation. So in those cases we need to be careful that how we need to check that whether any kind of a variations are being produced or not, right? So these things we need to take care, and definitely we can go for these kind of techniques, right? So this was all for today's lecture. So if you have any questions, you can ask your questions, and in the meanwhile I'll just post one assignment question also. And I see that some people, they are not responding to the assignment questions. Please, I told you earlier also that assignment questions are important. Do these questions, submit these questions, because the overall assessment in a class assessment is being done by these questions only, right? So please don't leave them as such. Just try to do them. And by this time you must have got the message also that for now the classes are online till further notice, and university is also being closed for the moment, so that because cases are increasing, right? So I really hope that you all are indoors, not going out, not moving out, because it is good to be safe, right? Okay, I have assigned the question. Maybe within 2 minutes or 1 minute, you'll be able to see. Submit them. The time is before 12 today. That means 12 p.m. So you are going to submit within 5 minutes, right? And we are going to discuss few answers that you have given. Just try to write in your own word what have you understood from this lecture. Just don't copy and paste. Right? Okay, I can see only 7 students turned in and rest still haven't submitted. Please hurry up. मुझे लगता है मैंने 12 बजे तक रखा था, उसके बाद late turn ins होगा आपका. Fine. So those who have submitted are Manaswini ने सबसे पहले submit किया था. Then we have Devanshi also, Nehal also, Tahiba also, Shivani also, right? So Shivani has written, right? She's written that it is a process through which a whole embryo development occurs from a single somatic cell, which are the cells that are not involved in the embryo development of a plant cells, that is a normal plant cells or a tissue. Okay. Then we have Tahiba. Tahiba has written, okay, a procedure in which a plant embryo is created artificially from a single somatic cell, which are not engaged in the formation of embryos like ordinary plant tissue. Okay. Then we have Shivani Mishra. Yes, Shivani, it is a process through which from a single somatic cell. Okay, the cells which are not involved in the embryo development. And we have a Manaswini also. Manaswini has, it is a process of regenerating non-zygotic embryos from a single cell or a group of cells, and they can be used to fully produce a plant. Okay, that's right. Then we have Shivani Mishra. Okay, so other students also, this is Shivani. Process through which a whole embryo development occurs from a single somatic cells which are not involved in embryo development of a plant, that is a normal plant cell. Okay. Okay, many students have turned in, but still many students are not there. Kshitij, I don't know, he's absent, I guess. Vivek Kumar Gupta not turned in, Yukta not turned in, युवराज also not turned in. युवराज पिछली बार भी 10 घंटे लेट टर्न इन किया था आपने. And we don't have Athoeba also, right? Okay, fine, students, please submit them. I'm again and again telling you this is important, right? So please, for attendance, switch on your cameras. Adhish Mishra. Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi.
Adish Mishra present.
Okay, Adish. Devanshi.
Present, ma'am.
Ishita.
Present, ma'am.
Janvi.
Present, ma'am.
Kshitij. क्षितिज, मैं आपकी एब्सेंट लगा दूंगी अगर आपने मेरी क्लास में अटेंडेंस नहीं बोली तो.
Present, ma'am.
मनस्विनी.
Present, ma'am.
Present, ma'am.
या नम्रता.
Present, ma'am.
नेहल.
Present, ma'am.
विपल.
Present, ma'am.
साहिबा.
Present, ma'am.
संजना.
Present, ma'am.
शक्ति.
Present, ma'am.
शिवानी.
Present, ma'am.
मानसवनी तो हो गया था ना. शिवानी के बाद कौन है? तन्वी.
Present, ma'am.
त्रिदेव.
Present, ma'am.
युक्ता. युवराज. Okay, Yuvraj, what happened? Why your mic is not working? Why you're not saying it? Huh? Please make sure that you take part in your attendance also. Your mic is also working properly, because for I don't know how long we are going to be online again. For at least for this month we'll be online, I guess, right? And Yukta, your mic is also not working. What happened to your mic, Yukta? I mean, why you guys are not replying? Okay, so in absentees, we have Vivek is absent. We have Athoiba is absent. Vivek and Athoiba. Two people are, and anyways, I'll download the attendance also. Please tell all the students to attend the classes regularly. Like Athoiba, I guess he was absent last week also. So Adhish and any other student who are in touch with Athoiba, let him know that classes to attend the classes, right? Okay, thank you so much. So I'll see you tomorrow in the next class.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.